Today we will talk about DNA and chromosome structure. First, let's talk about the basics. What does DNA look like? DNA is constructed by a bunch of monomers called a nucleotide. A nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. When connected, the nucleotides create the sugar-phosphate backbone of DNA. Two strands of the DNA come together to form a double helix structure. It does this by identifying the complementary base pair on the other strand of DNA. A goes with T and G goes with C. The base pairs are held by hydrogen bonds. Now that the basics about DNA are covered, let's go into DNA packing. When does DNA packing take place? DNA packing occurs during cell division. The chromatin gets packed together during the mitotic phase. The chromatin is then transformed into a chromosome. The first step of DNA packing is when small proteins called histones attach to the DNA double helix. 8 histones come together and DNA is wrapped around them. This bead-like structure is called a nucleosome. Next, the nucleosomes, or bead-like string, come together and form a tight helical fiber. Then the fiber turns into a supercoil and eventually packs tightly together into a chromosome. Now we can talk about chromosome structure. During the S phase of interphase, the chromatin is duplicated. The duplicated strands are coiled just the same as the original strands. Because the strands are so close together, the two chromosomes formed can be bound at the centromere. The duplicated and original chromosomes are now called sister chromatids. They are only referred to as sister chromatids when they are connected at the centromere. When separate, they are chromosomes. How does the structure of DNA affect gene regulation? Tightly packed DNA does not get expressed while loosely bound DNA does. This is due to methylation and acetylation. Gene regulation is what controls which genes are expressed and which ones are not. Methylation is when the genes are not being transcribed, and acetylation is when genes are being transcribed. A methyl group added to DNA turns off the gene. This happens because the DNA becomes tightly packed and prevents transcription. On the other hand, an acetyl group attached to a histone changes its shape and grips the DNA less tightly. As a result, transcription can occur. A prime example of this is how each organ and every part of the body are developed in the embryo. This process is necessary for cellular differentiation in an embryo. Each cell has the same DNA, but based on methylation and acetylation, cells get differentiated into skin, brains, eyes, etc. What factors of our environment can affect our DNA? Well, radiation and chemicals are to blame. These factors can cause mutations, and mutations are changes in amino acid sequences. Radiation and chemicals may cause a deletion or other change in amino acid sequences, which results in abnormal protein synthesis. Cancer cells are an excellent example of mutations. A change in an amino acid results in disrupted checkpoints in the cell cycle, which produces abnormal production of cells. Now for some fun facts for the day. If you unwrap all the DNA in all of your cells, you would reach the moon. Also, there are approximately 3 billion chemical letters, otherwise known as bases, in the DNA code in every cell of your body. And one last fact for the day. You share about 50% of your DNA with a banana. Thank you for watching, and I hope this was informational.
Created using Powtoon.